In this video, Brad Pitt will be helping us to explain the structure of DNA. Okay, so let's open this box of secrets and let's just get into it. So basically, we know that just like Brad Pitt, right? Just like Brad Pitt, we are made up of trillions of cells, right? Trillions of cells. If our cells are working well, then we are working well. If our cells are dying, then we are dying, okay? So we know we're made up of these cells, and we know cells have a lot of organelles, right? A lot of organelles, like mitochondria, like the nucleus, okay? So we're gonna be focusing specifically in this video on the nucleus, because that is where our DNA is, right? If we zoom into the nucleus, that is where we can find our genetic material, our DNA. Now, what exactly does DNA stand for? DNA, okay, stands for deoxyribonucleic acid. Deoxyribonucleic acid. And later on in this video, you'll see why this word is named this way, okay, why we call it this way. Now, what exactly is DNA? A good way to think of what DNA is like an instruction manual, okay, an instruction manual for your body. Like, think about this. When you order online a product, such as, let's say, a gym set, okay, or a bicycle, if you buy this product and it gets delivered to your house, most likely you will get an instruction manual with it, right? This instruction manual helps us so that we can read on how to build this particular thing, okay? So that's basically what DNA is. It's our instruction manual. Our body, our cells will read this DNA and make sure that they do what the DNA says, okay? They make sure that they do what the instruction manual says. And if it follows the instruction manual well, we should be all good, okay? Now, an interesting thing to bear in mind is that DNA from a single cell, so if we take one body cell, and we take the DNA out of that nucleus, it will be 2 meters long. So that's insane considering how small one cell is. To emphasize this, if we take the DNA from all the cells in our body, this will span the diameter twice of the solar system. So that's crazy considering how small, how small a cell is for how much DNA it has. Now, so one question we can come from this is, how is it possible that DNA can fit inside our nucleus if it's so long? So this is something that we'll answer a bit later on in this video. Now, anyways, let's go on to the next thing. As I said, DNA is like an instruction manual, right? So our cells will read it and follow what it says. Now think about the English language, right? The English language has a lot of, it has a whole alphabet, right, with like 26 letters, right? So if we take these letters and put them in a certain order, we can create a word, right? So let's say we took these 6 letters and made a word called silent. Now if we take these exact letters from this word and rearrange them, we can come up with a new word, okay? So such as listen. Now if we take a bunch of words and put them together, we can form what? We can form a story, such as Harry Potter, right? We can form a story. Now, let's say we take these words from Harry Potter and rearrange them in a certain way, then we can create what? A new story, right? Now, what am I trying to get at? Just like a story, just like English language has these letters that can create words, that can create stories, our DNA is also made up of letters, okay? Our DNA is also made up of letters, a sort of an alphabet. Only this alphabet only has 4 letters, C, A, G, and T. That is the 4 letters that make up the DNA alphabet, okay? And instead of calling these letters like we do in the English language, we call them bases, okay? So bases is the equivalent of letters. Now, just like how we can put these in a certain order, just how we can put words, letters in a certain order to create a word, and we can put a bunch of words together to create a story, we can do the same with the DNA letters. Depending on the order or the sequence of that these letters are put in inside our DNA, that will create a different organism, right? So a different set of sequence will create a different organism. Now, just like if you misspell a word, suddenly that word doesn't make sense. If our DNA is wrong and something is wrong with our DNA, such as a mutation or a genetic issue, then you will have a disease, right? Because just like that word didn't make sense, now some part about you won't make sense or won't be correct, okay? So this is just related to this idea of rearrangement, okay? If our letters are rearranged, we will be a different organism, we'll have different physical traits, such as different hair color or things like that, and in worst cases, we could have a mutation in which we will have a specific like issue, okay? So, this brings me to the next part. What are all these letters I've been talking about, right? Where are they? Where are they exactly? So I'm saying that just like the English language has letters, our DNA is made up of these 4 letters, this alphabet, okay? Now, where are they? Where can we see them? Okay, where can we see them? So if we look, right, we look at Brad Pitt, we know he's made up of cells. If we look inside this nucleus, we're going to see DNA. And DNA looks like a helix, kind of like this. Now, notice all these colors here. These are the alphabet. So I didn't put letters on here. I didn't put any A, G, C, or T's, but I color-coded them just like this, okay? So this is where the code is, okay? So depending on how these letters are arranged and how they're put, this will code for something different. This will make you unique compared to anyone else, okay? So this is the instruction manual. Now, I want you to look at this and notice some things. What do you notice? So again, you notice only 4 colors, right? That's because there's only 4 letters, only 4 bases. What else do you notice? Maybe you notice that the blue is always with the green. Okay, the blue is always right next to the green, and the red is always right next to the orange. So this is also something important that we're going to get to later. And maybe you notice even something else, that in between the blue and the green, there's 3 bonds, whereas in between the red and the orange, there's 2 bonds. Okay, so maybe you're noticing all these things, and don't worry, we're going to talk about them. But for now, I quickly want to come back to this point again. Remember how I said DNA, a cell, the DNA from a single cell is 2 meters long, right? How on earth does that much DNA fit into a nucleus? Let me show you. This is something important that you need to know. So let's say we take the DNA, right? And we basically what happens is this DNA, the only way it can fit into here is if it coils up really, really tightly. So imagine a DNA is like a rope, okay? DNA is like a rope. And you take this rope. You take this rope and you take these pink things, okay? So these pink things represent like a finger or any kind of object. So normally when you try and roll something up, you got to roll it up around something, right? Like your finger, if you roll this this rope up around your finger to make it nice and small, nice and tight. That's the same thing here. These pink things here are called histones. Let me show you. They're called histones, kind of like little stones, okay? And they serve as the little object on which we're going to coil this DNA. So we're going to coil it around these histones. There are 9 of them. If you count 1, 2, 3, 4, 5, 6, 7, 8, 9, there's 9 of them. Okay, so those pink things are the histones. Now, after we coil the DNA around these histones to make it tighter, now we call this the combination of the histones and the DNA. We call another name. We call it nucleosome. Okay, so this right here is one nucleosome, the combination of the DNA and the histones. Now, this is not where it stops. So we made it a bit tighter now. We wrapped around the DNA around these histones to form a nucleosome. But now, you have to bear in mind that we're not only using one histone. This happens because our DNA is so long, so we have to use many histones. So what happens is, this same process will repeat with a bunch of histones. Let me show you. So basically, you can see here, each of these here, so this one right here is one entire nucleosome. One entire nucleosome. This is another nucleosome. This is another nucleosome. So the point is, you take a piece of DNA, you rotate it around the histone, and then you take another piece of DNA along the line and rotate it around another histone, and then the next piece of DNA rotate around that histone. So basically, a lot of histones are used. A lot of nucleosomes are formed. And after a while, now we have all the DNA coiled up, and now we got to coil it even more. So we take these histones and we coil them around each other. Okay, we take these nucleosomes and we coil them around each other. So you can see all these nucleosomes are now being coiled here around each other. And then this continues even more. The coiling continues even more. Okay, so we keep coiling, keep coiling, keep coiling until it gets basically so tight that it takes the shape of a chromosome. Okay, so you notice here, we coiled this DNA so tight to form a nucleosome. We coiled these nucleosomes even tighter, and we kept coiling and coiling and coiling until it was something called supercoiled into the shape of a chromosome. And we know we have like 46 chromosomes inside here. Okay, so that is how our DNA is 2 meters worth of DNA manages to fit inside our nucleus. Okay, so that's important. That's one key thing we have to cover. Remember these two words, histones and nucleosome. Okay, very important. Now let's go back a little bit. Okay, we're going to look now at this DNA here, because we started talking about these codes, these alphabet letters that are going to code like a storybook, right? That your cells will read and use to make you look the way you do, and so on. Now let's look a bit more clear. So we're focusing on a piece of DNA, and what I did was I made a diagram here that we opened it up. So because normally DNA is in a helix shape like this, right? So I opened a bit of it up so we can see clearly everything that's going on. Now if we use what do you call this again? A magnifying glass, and we look real close. For example, in this magnifying glass, we're looking at this part here. Let me show you. More or less looking at like this area, okay? We're looking here, okay, because we want to see what's really going on. Because this image here is a bit simplified. So we're going to look a bit closer to see what's really going on. And we notice when we look closer, it's a bit more complicated than we thought, okay? We notice that this area here, this dark area here, actually is made up of little molecules. This red one here and this little purple one here, okay? So what's going on in this dark area here and here is actually little structures making it up, okay? And you'll learn later we call this the backbone, but don't worry about that now, we'll get to that. And then we have these letters here, right? These letters, which actually have a specific shape. They're not just like a little rectangle, they have a specific shape, okay? So very important. And don't worry, we're gonna look at this now in detail, make sure we get everything. But the point is, you can see that the DNA essentially has 2 halves, right? 2 halves. This is 1 half. Okay, we call this one half of a DNA strand, right? And then it's reflected as the other half. This is the other half, right? So there are essentially 2 strands, okay? 2 strands. One strand of letters going down here, and another strand of letters going down here, right? Now, the smallest thing making up the DNA would be one of these, okay? You can see this unit repeats. Here's the unit, it repeats again and again and again, it repeats, right? So where is that on this diagram? On this diagram, 1 unit is this. This is 1 unit. What do we call 1 unit like this? What do we call 1 unit like this? We call it a nucleotide, okay? Let's look at it on the next page. Here we have a nucleotide. We're zooming into this single nucleotide here, okay? So we know this is the smallest thing that makes up DNA. Let's cover some things that we need to know. So first of all, let's label some names. So this group right here, this round one, is called the phosphate group, okay? Phosphate group. Then we have this kind of group here called deoxyribose. Don't worry, we're going to talk about it just now. And then here is the base, okay? I made this colorful right here because remember, the base is the letter, and it can be any one of 4. Any one of these 4 colors, right? So right now we're looking at the pink one, but it can be any. It can be any. This one can be substituted for any of the other bases. Okay, so this is the 3 basic structures that a nucleotide is made of: a phosphate, deoxyribose, and a base. By the way, the deoxyribose, bear this name in mind. Remember, the name of DNA is deoxyribonucleic acid. So you can see the beginning part, deoxyribo, is called deoxyribo because one of the components of DNA in the nucleotide is this deoxyribo sugar, okay? Now, before we move on, I want to quickly do something. So look at this deoxyribose. In reality, how this looks like. So you think now, oh, look at all these shapes. So what's really going on here? What's really going on here is this is a picture from a textbook. Now, we're making a shape here, but in reality, the real shape is that each of these corners here is a carbon. So notice this corner, there's a carbon. This is a carbon. Here's another carbon. Here's another carbon. And here's a carbon. And up here is an oxygen. So these corners represent an element. And then these sides represent a bond. Okay, so you can see here carbon 1 is bonded to carbon 2. So if we have here carbon 1, carbon 2, this is the bond in between carbon 1 and carbon 2. I'll show you clearly. So carbon 1, and then up here is the oxygen, okay? So very important. So when I draw these shapes, I'm not just drawing a random shape. It resembles what the real molecular structure is, okay? It resembles what the real molecular structure is. Okay, now, what else do we got here? Remember how we looked at this, right? And we saw that these two, the phosphate and the deoxyribose, is inside this part here, okay? And I called it the backbone, okay? So let's label that here. So this whole nucleotide, if we consider just the phosphate and the deoxyribose, so these two, we call this the backbone, okay? We give it a name, call it the backbone. And it basically represents the same function as your backbone. Your backbone is your spine, right? Without your spine, you'd be jelly and you'd be on the floor. You wouldn't have any good structure or shape at all, right? So that's the same thing for a nucleotide. These two, the phosphate and the deoxyribose, functions as this backbone, okay? The structure, keeping DNA in a good structure, okay? The letter is the actual code. The base is the actual code that your cell is going to read, but this is just a backbone. It keeps structure, okay? Now, notice how the deoxyribose is bounded to a base, but also to a phosphate. What kind of bond is this? Okay, so we give this bond a name. We call it a covalent bond, a covalent bond. So the bond between all of these here is a covalent bond. It's a really strong bond. Bear this in mind because later we're going to learn about another kind of bond. Okay, now, you know how if you look at a human, you can look at them and say, that's their head, that's their toe. So we also need to learn that for this molecule. Okay, so what's the top of this molecule? We know it's up here, right? And what's the bottom? Down here, right? So they give it a name. So instead of calling it top and bottom, they call it 5' end and 3' end. Which side here of this molecule, the top or the bottom, do you think is the 5' end? The top, because 5 is the highest, okay? It's the highest number. It's up here. It's near the top. So the top will be called the 5' end, okay? And maybe you know about Logan Paul. He's creating prime, so why not put prime here? So 5' end is the top of the nucleotide, whereas 3 is the bottom here, so we call the bottom end the 3' end, okay? So that's it for what we need to know about a nucleotide, okay? We need to know these different things that make it up. We need to know what's the backbone. We need to know about this bond, and we need to know the top and the bottom, also known as the 5' end and the 3' end, because that'll matter a bit more later. Okay, so let's keep moving. So remember how we said that there's 4 letters in the DNA alphabet, right? A, T, C, and G. Here they are. Okay, so we got the G is guanine, the C is cytosine, the A is adenine, and the T is thymine. So these are the entire alphabet of the DNA. Okay, now notice how guanine and adenine look similar in structure. Okay, they have a similar structure, whereas cytosine and thymine have a similar structure. So we call cytosine and thymine, we classified under a group called pyrimidines. Okay, whereas adenine and guanine we classify under a group called purines. This is easy to remember because pyrimidine has a Y in it, and both cytosine and thymine are pyrimidines, and they also happen to have Ys in them. And if you can remember these two are pyrimidines, then the remaining two are purines, okay? So this is not too important of a thing, but technically it's in your book, so you need to kind of be aware of this at least, okay? Now, let's get a bit more into the final bits here, the final bits. Okay. So we learned now how individual nucleotides are like and what we need to know about them. But we saw that here, the nucleotides are bounded to each other. They are attached to each other. This one is attached to the one above, but also to the one below. This one is also attached to one to its right. Okay, so they're all attaching to each other. They're not free. They're not floating around free. Okay, so how do we attach them to each other? How do we do that? So let's take it. If you look at a nucleotide, what links them together is the third carbon. So this carbon here, remember carbon 1 was here, carbon 2 was here. So the third carbon on the deoxyribose will link to the phosphate of the nucleotide below. Okay, so that's how they link. So now we managed to link them above each other. Okay, so This one links to that one, links to that one. But now we know that they also need to link to one on its right, because I said, remember, that DNA has two strands, one strand going down here and another strand going down here, and they're linked to each other with these bonds, okay? So how does those bonds happen? So, unfortunately, for some reason on Earth, the opposite strand is upside down, okay? The opposite strand, so these two strands, this strand and this strand is upside down from each other. So you can see this one has the 5' end on the top, remember? 1, 2, 3, 4, 5, the 5', so this is the 5' end, and then here is the 3, so this is the 3' end. So this strand of DNA, the top is this side and the bottom is this side. Whereas for the opposite side, so the opposite strand, if we follow the same rule, so this is the first carbon, second carbon, third carbon, the third carbon is actually, the 3' end is actually the opposite side, and the 5' end is on the top side. So this is one thing you need to understand, that the two strands are anti-parallel. Okay, here's the word, anti-parallel. Parallel meaning they're next to each other, and anti means they're going the opposite direction. Okay, so they are anti-parallel. Okay, that's very important to understand. So they're just the same thing, just rotated the opposite way. Now, importantly, out of these four nucleotides, okay, A will always bind with T, and C will always bind with G. How can we remember this? So if you haven't noticed, on my picture here, you'll notice, like we talked about before, green always binds with blue, and orange always binds with red. That's because A always binds with T, and C always binds with G. It will never be G binding with A or anything like that. They always bind to the same nucleotide. So we call this property complementary base pairs. The base pairs complement each other, okay? So how can we remember this? A and T are always together. So A and T will always bind together. Adenine and thymine will always bind to each other. So here, right here, this one is thymine, okay? Thymine, and it's always going to bind to adenine. So we're going to have a bond here, okay? We're going to have a bond. A and T will always bind to each other. Now remember, one thing you need to remember is A and T always have two bonds, okay? And again, you can remember that the same way. Always have two bonds. A and T always have two bonds, okay? Whereas C and G, okay, always have three bonds. And what do we call these bonds? We call these bonds hydrogen bonds. So remember before, these ones we called covalent bonds. They were very strong. Now, linking side by side, these two DNA strands, linking them side by side, these are hydrogen bonds, and they are weaker. Think about the beginning of this word, hide. Okay, that's like hiding in the closet, right? Hiding is a sign of weakness, okay? Now, hydrogen bonds are weaker than covalent bonds, so they are more easily to be broken. So they're hiding bonds, they're weaker, okay? And then lastly, we have here this one. That's also going to be 3 bonds. Okay, 3 bonds. Now, notice how, remember we learned about pyrimidines and purines. You can see here that one purine always binds with pyrimidine, because look, the G binds with C. So one purine binds with the pyrimidine, and the A binds with the T. So the purine binds with the pyrimidine. So why? Why does one big one always bind with one small one? Why does one purine always bind with a pyrimidine? This is because of a size factor. Okay, if the big one bonded with the big one, then the DNA would be really wide. Okay, and if the small one bound with the small one, then some piece of the DNA will be very narrow. So having it like this, one big one bound with a small one, makes it always equal in width. Okay, and this makes the DNA very, very stable, very, very structured. Okay, so that's also very important to remember. So, okay, I think this is, oh yeah, we forgot to link here. Remember here, the third one, 1, 2, 3 will bind to the phosphate. Same as here, 1, 2, 3 binds to the phosphate. And one good way to remember that these bonds, how these bonds work, is that each component, so this, the deoxyribose, and this one always binds to at least 2 other components. So if we look at this base here, it is always binding to another base and the deoxyribose. So it's binding to at least two things. This ribose here is binding to at least two things. It's binding to the phosphate, the base, and another phosphate, okay? And this phosphate is always binding two things. Always the ribose and then the other ribose, okay? So if you ever get confused on how to, for example, you had this bond here and you forgot how you link these two nucleotides and you're like, oh, it's gotta be from here to there. This is wrong, and it's easy to remember because right now, this phosphate isn't making at least two bonds. And I said each component needs to bond to at least two things. And right now it's only binding to one thing. So you can fix that by doing this, right? So that is it. That is all you need to know for the structure of DNA, okay? Because in the next video, we will talk about how your body actually reads this DNA. Because we talked about that it's an instruction manual, but how exactly does your body read this DNA? How does it make use of it? How does it read this DNA to make you the way you are? Okay, so that's the next video on transcription and translation and all that.